Hi everyone. In this presentation, I'd like to introduce you to a commonly used tool in biotechnology and molecular biology. It's called PCR, or polymerase chain reaction. PCR is a technique that was developed by an American biochemist and now Nobel laureate named Kary Mullis. In 1983, Mullis was working on a couple of different problems. He was trying to figure out how to increase demand for a product that is lab-made called oligonucleotides, and he was trying to come up with a technique that would allow you to determine whether or not human cells, like those you'd find in a fetus, could contain unfortunate or detrimental mutations. His idea was that if you could find those mutations early enough, you could warn new parents that their child might face significant challenges in life, and they could, as a result, make the proper preparations and adjustments. At the time, finding mutations involved taking genomic DNA from human cells, extracting it, cutting it up into thousands of pieces, and then inserting those pieces into bacteria for study. It was a very long process, taking weeks and sometimes even months. With Mullis's discovery of PCR, however, scientists no longer needed to cut up DNA genomes or insert the resulting fragments into bacteria that then had to be grown up in large enough amounts before you could even determine what a mutation was in the DNA. So the experiments went from taking lots and lots of reagents or chemicals, lots and lots of plastic tubes and things like that. It went from being a hugely resource intensive experiment to being an experiment that could be done in one test tube. The same work that took days or weeks or months, it could be done in an hour. Well, maybe an hour is an exaggeration. It could be done in a few hours, maybe a couple of days. Now, Mullis's experiment, his discovery, like many great American discoveries, came through ingenuity and time out in the garage. That's right, the first PCR experiments were done using water baths and other basic lab equipment in a garage in California. Nowadays, PCR is a basic technique that any skilled molecular biologist can do blindfolded with one arm tied behind their back. Okay, maybe that's an exaggeration, but it's not uncommon for this technique to be taught in high schools around the nation. Let's talk about a few ways that PCR is used in research today. In DNA profiling, a biological specimen like blood or tissue, bone, or maybe cells from a cheek swab is matched to an individual. Most of our DNA is the same, but some sections are highly variable and called polymorphic. We inherit a unique combination of polymorphisms from our parents, and they can be analyzed to reveal family relationships, like what you see in paternity testing, or it's sometimes used to identify victims of a disaster or war. For example, the Army uses this technique to identify soldiers that we've lost on the battlefield. DNA profiling can also be used to identify the perpetrator of a crime. You might have heard of this somewhat recently, as this is how the Golden State Murderer was identified in 2018. Sometimes PCR is used to identify microorganisms. For example, blood specimens that were donated to the Red Cross may be tested for HIV sequences, since they can be detected before the body makes antibodies to the virus. This technique may also be employed to look for HIV in babies born to infected mothers. Now, when scientists know something about the genetic mutations or the locations of genetic mutations that lead to disease, PCR can be used to probe genomic DNA from potential people who have these disorders, okay, and those mutations can be identified. PCR is also used to distinguish and classify newly discovered organisms in a process called DNA barcoding. But what about genetic engineering? In genetic engineering, DNA genes are copied and then inserted into the DNA of embryos or plants to change the characteristics of the resulting organisms. Through this technique, a cow, for example, may be able to produce new vitamins or proteins in their milk, or plants can be given genes that make them resistant to specific insects or drought. One thing I'll say here is that this kind of work isn't as easy as it sounds, but PCR is a part of it. Finally, PCR can be used to add random or specific mutations to genes in order to understand and determine their function. I used to do this kind of work regularly when I worked in a lab. PCR is a chemical reaction in which a strand of DNA, called a template, is copied or amplified billions of times in a matter of hours. The resulting DNAs, like you see here, are called amplicons. The amplicons can be analyzed for the presence of mutations, for size differences, or they can be inserted into other DNAs like plasmids for study. I hope you're asking yourself, well, how do we do this? If you consider that a genome can have hundreds of thousands to billions of base pairs, remember a base pair is something like an A bonded to a T or a C bonded to a G. You can imagine that it would be time-consuming and difficult to analyze the entire genome of a cell in only a few hours. Instead of making billions of copies of the entire genome for study, PCR only amplifies a specific stretch of DNA, like part of a gene, for example. This stretch is defined by two small pieces of DNA, highlighted in red here, called oligonucleotides. Nowadays, they're called DNA primers. These are the molecules that Kary Mullis's lab was synthesizing. The oligonucleotide primers have complementary sequences to the template DNA strand. So this is my template. In PCR, I'm going to separate it into single-strand pieces like this, and then the DNA nucleotides, the DNA primers will bind because they have complementary sequences to the template DNA. To these DNAs, we add a heat-stable DNA polymerase. You see, during this chemical reaction, we're going to cycle all of these DNAs between temperatures that are close to boiling and temperatures that are much warmer than that of our own body. So it's important that the enzyme be able to withstand high temperatures without breaking down. That's why we use polymerases that are derived from thermophilic and hyperthermophilic organisms. Now, if we're going to make new DNAs, we need nucleotides. Deoxynucleosides, called DNTPs, are the A's, T's, and G's that will be incorporated into new copies of our DNA sequence. Next, buffers are added that provide the proper pH and ionic environment for the polymerase to work. And then we add a little bit of water just to hold everything together, dilute the buffer, and have the correct volume for our reaction. The template DNA, primers, DNA polymerase, DNTPs, buffer, and water are all added to a small PCR tube that looks like this. And then that's placed into a machine like this. This is called a PCR thermocycler. The thermocycler, like its name suggests, cycles the chemical reactions at different temperatures, about 30 to 40 times, in order to amplify our template DNA. A typical PCR reaction consists of 3 steps that are repeated 20 to 40 times. The first step is called denaturation. Here, the thermocycler raises the temperature of a PCR reaction to about 94 to 96 degrees Celsius. This is hot enough to break the hydrogen bonds holding double-stranded DNA together. As a result, the strands of our double-stranded DNA template separate, as you see here. During the second step, which is called annealing, the temperature of our PCR reaction is lowered to about 68 degrees Celsius. This allows the DNA primers, or the oligonucleotides represented by these red Lego bricks here, this allows them to bond to our template DNA according to complementary base pairing rules. So A is going to pair with T and C is going to pair with G. If we were to blow this up, we might see something that looks like this. So the black letters represent the DNA template sequence, and then the orange letters here and the red letters here represent our primers. So I hope you can see that the A's pairs with T and the C pairs with G. This is how the primer knows where to bind, and also this marks the ends of the DNA sequence that we want to copy, that we want to amplify. During the third step of PCR, which is called elongation, the temperature of the PCR reaction is raised to approximately 72 degrees Celsius. This temperature is determined by whatever DNA polymerase we decide to use for this experiment. In this step, DNA polymerase binds to the 3' end of the primer here, which, by the way, corresponds to the 3' end of our template strand, and then it reads the nucleotides in a 5' direction and adds the complementary base opposite. This happens on both strands. So the primer, as well as all of the new nucleotides, become part of our new DNA strand. So what you can see depicted here is exactly that. Here's my template DNA in green, here's the primer in red, then the DNA polymerase reads the nucleotides on the template and inserts the complementary base opposite. So the blue here represents our new DNA. This happens over and over and over again, generating billions of copies of our desired DNA sequence. PCR reactions may be analyzed by agarose gel electrophoresis. Agarose is a polysaccharide extracted from seaweed. When it is heated with a buffer, it forms a viscous liquid. As it cools, it forms a dense gel matrix, somewhat like gelatin, if you've ever made jello. In the lab, we pour cooled but still molten agarose into a mold and allow it to cool slightly and solidify. Microscopically, the agarose molecules gel together like this and form a matrix. So imagine a matrix like this occurring all throughout this slab of agarose that we have on the right side of the slide. We apply DNA to small depressions called wells at one end of the gel. Then we enhance the negative charge on the DNA by placing a negative electrode nearby. As you know, opposite charges attract. So we place a positively charged electrode at the opposite end of the gel. So what happens is that once we start this reaction, DNA that starts out in the wells up here begins to migrate through the gel, through this matrix that you see like this, okay, migrates through the matrix towards the positive electrode. This will separate DNAs of different sizes because longer DNAs take more time to wiggle through the gel matrix than smaller ones. We can cut these DNAs out of the gel and then analyze them further or insert them into plasmids or other larger DNAs. If the goal of an experiment is to know the order of the A's, T's, C's, and G's of a DNA fragment, then PCR followed by capillary DNA sequencing might be used. In this type of experiment, PCR is performed, but instead of using all normal nucleotides, a small percentage of fluorescently labeled defective nucleotides, say about 1%, are added to the PCR reaction. By defective, I mean that these nucleotides terminate DNA synthesis, and by fluorescently labeled, I mean that these nucleotides have a little bit of extra chemistry that allows them to glow when a specific wavelength of light is shown on them. So defective A's may glow green, C's may glow blue, defective G's may glow yellow, and defective T's, well, they might glow red. Again, these defective nucleotides are mixed with an overwhelming number of normal nucleotides. This means that as the PCR reaction runs, normal nucleotides will be added to new DNAs by DNA polymerase, but occasionally a defective nucleotide gets added, and when that happens, DNA synthesis for that strand stops. To make this easier to understand, let's take a look at an instance where defective Gs are included in a PCR reaction. Here's a DNA fragment that we would like to have the DNA sequence for. Remember that during PCR, primers bind the ends of our DNAs, and then DNA polymerase binds the end of the primer. And it reads the nucleotides in a 3' to 5' direction, one by one, and it inserts the complementary base opposite. So where it sees an A, like this one, it puts in a T. Where it sees a G like this one, it puts in a C, and so on and so forth going down the DNA strand. As long as our reaction has only normal nucleotides, we should wind up with DNA fragments that are the same length. During a sequencing run, however, remember that 1% of our nucleotides are the defective fluorescently labeled type, so DNA synthesis will stop whenever one of those defective nucleotides are inserted. So a PCR cycle with this DNA might return a complementary DNA sequence that looks like this, where the DNA is truncated prematurely because a defective nucleotide was inserted. Alternatively, maybe we get a sequence that looks like this, or this, or this, or this. Here, that defective nucleotide is being inserted randomly along the sequence, and wherever it goes in, DNA synthesis stops. In my example, only defective guanines were included in the PCR reaction. Nowadays, a PCR reaction will contain some ratio of defective adenines, thymines, cytosines, and guanines. So we wind up with a lot of DNA fragments that have been prematurely truncated or shortened by the insertion of defective, fluorescently labeled nucleotides, kind of like what you see here in this figure from Sigma Aldrich. This mix of fragments is separated by capillary electrophoresis. We're doing the same thing as in agarose gel electrophoresis, but we do it in a capillary tube instead. When a laser beam is shown through the glass of the capillary tube, it causes the defective nucleotides to glow, and the order of the nucleotides can be read by a machine. If you're interested, Eric Lander gives an excellent description of Sanger DNA sequencing as part of his 7.01 SC Fundamentals of Biology lectures from MIT. You can find some of these lectures on YouTube, and if you're a biology major, I highly recommend checking them out. Thank you.